Hey, how you doing? Today's video is going to be about enzymes. So let's get started. Alright, so we're going to consider enzymes under 4 different videos. The first of which includes your introduction, nomenclature and classification of enzymes, factors affecting enzyme activity, and active site of enzymes. So let's begin with a short introduction about enzymes. Enzymes are biocatalysts formed by living cells. Now, they are protein in nature except for RNA, which forms ribozyme. So RNA is the only enzyme that is not a protein. They are colloids. They are thermolabile. Now, thermolabile means that they are sensitive to heat. That is, on heating, they are destroyed, they are degraded. Which is why we say that enzymes have an optimum temperature at which they function. We'll get into that later. And another property of enzymes is that they are highly specific. They're highly specific about the substrate on which they react or the reaction that they perform. Again, details of this we'll explain to you later. Now, there are some terms that I would like you to know before we get into the details of enzymes. Right. What are they? You have apoenzyme. Apoenzyme is the protein part of an enzyme. Now, I just said that enzymes are proteins. So, why am I saying protein part? Because enzyme is formed of a protein part along with a non-protein part which helps in its functioning. Protein part is called apoenzyme. The non-protein part is called coenzyme. Apoenzyme plus coenzyme together form holoenzyme, which is your fully functional enzyme. Now, coenzyme is of two types. It is cofactor or prosthetic group. So, when the coenzyme is tightly or covalently bound to the apoenzyme, it is called prosthetic group. It is not easily separated. When it is less tightly or loosely bound, non-covalently bound to the apoenzyme, it is called cofactor. So now that we've got these terms, let's dive deeper into the topic. Alright, moving on, let's discuss nomenclature and classification of enzymes. So initially, enzymes we used to use common names or trivial names for enzymes like trypsin, chymotrypsin, etc. But that doesn't explain anything about the substrate on which an enzyme reacts or the reaction that it performs, right? So it sort of became confusing, which is why a union was formed, that is IUB, the International Union of Biochemistry, which decided to fix a nomenclature for enzymes. Alright, so an EC number, that is Enzyme Commission, alright, EC stands for Enzyme Commission, was allotted to every single enzyme. Now, this EC number consists of 4 digits, alright, and each digit tells us something about the enzyme. So, your EC number, the first digit is, it gives you the class of the enzyme. Enzymes are divided into 6 classes, and that forms the classification basis for enzymes. We'll come to that in a minute. Alright, the second number gives you the subclass. The third number gives you the subclass, and the fourth number gives you the serial number. Alright, now altogether this tells you about the substrate, the class of the enzyme, the substrate on which it reacts, the reaction that it performs. Alright, it gives all these details. Of course, this is not generally used. Not everyone would be able to figure out just from the EC number what the enzyme was. So along with the trivial names, this IUB classification is also used. Alright, so moving on to the class. There are 6 classes of enzymes. They are oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. a mnemonic to remember it is OTHLIL. Remember this, the order is extremely important. Alright. So let's look into each of these. Oxidoreductase. The name tells you itself, it's involved in oxidation reduction reactions. So in these kind of reactions, what happens is, say you have an AH2 plus B. Alright. What you'll get is A plus BH2. One substrate gets oxidized and the other gets reduced, right? Transferase means there is a transfer of a particular atom or a group from one substrate molecule to another. Alright. Then we have hydrolase. Hydrolase means water molecule is used. So you have A plus B plus say H2O. Under your enzyme action, you will get AH plus BOH, right? Then we have lyase. Lyase is involves the cleavage of a already formed bond, alright, and atom elimination. So that is the function of lyase. Then we have isomerase. Isomerase is involved in isomerization reactions. We've seen many of those. Then we have ligase. Ligase is involved in joining of molecules and formation or synthesis of a new bond. Alright. Now we have examples of each of these. Oxidoreductase has an example, alcohol dehydrogenase. Transferase example is hexokinase. It transfers phosphate group. Then we have hydrolase, example is lipase. We have lyase, example aldolase. We have isomerase, example is retinol isomerase. And we have ligase, that's succinate thiokinase. Alright. Alright, let's move on to the factors affecting enzyme activity. How would the reaction proceed? Okay, we have a substrate which is acted on by an enzyme to form a product, right? So what would the factors affecting this enzyme's activity be? The concentration of the substrate, the concentration of your enzyme, the concentration of the product, the temperature at which this reaction is occurring, the pH at which the reaction occurs, and the time taken to complete this reaction, right? So let's look into each of these now. First, we look at enzyme concentration. Now, if your enzyme concentration increases, the velocity of the enzyme reaction also increases, right? There is a direct relation between the two. It is directly proportional. Obviously, now if your enzyme molecules increase, there are more molecules of the enzyme to bind with the substrate. So your velocity of the reaction will increase, right? We move on to substrate concentration. Now, before we start with the graphs of explaining substrate concentration, there is a term that you must understand. That is Km. What is Km? Km stands for Michaelis-Menten constant, also called Briggs-Haldane constant, right? What is Km? Km is the concentration of the substrate at which the reaction reaches half of the maximum velocity, okay? Now, Km, the normal value of Km ranges between 10 raised to minus 5 to 10 raised to minus 2 moles, right? Now, your Km is specific to an enzyme. It is sort of the fingerprint of an enzyme, and it remains constant for any given enzyme. Alright. What is the meaning, what is the significance of Km? Km is, like I told you, it is the substrate. So now if your Km is low, it means that the substrate amount is low for you to reach half the maximum velocity, right? Which means that the enzyme has a high affinity for your substrate because you're reaching half Vmax with a low concentration of your substrate, right? So now that we've understood Km, let's look at the two graphs. The first graph plots substrate concentration against the velocity of the reaction, okay? It is a rectangular hyperbola, as you can see. It is not directly proportionate. Why? Because here we are assuming that your enzyme concentration is constant. So that gives you a limitation. Once your maximum enzyme concentration is reached, that is, your substrate has bound with all the present enzyme, further increasing your substrate is not going to change anything, right? Because your enzyme is already bound. There is no more enzyme remaining to bind with substrate. So initially it increases, but after a particular point, that is at Vmax, when your entire enzyme is consumed or bound rather, it reaches a plateau phase. Alright, so that is your rectangular hyperbola. You can see here that at half Vmax, your corresponding substrate concentration is Km, as we discussed just now. Now there is another graph to explain this, which is called Lineweaver-Burke double reciprocal. Okay. Now that is so basically what you're plotting is the reciprocal of substrate, concentration with the reciprocal of velocity of reaction, right? So what we see here is that your Km value, what we're going to get is 1 upon Km, right? And since it is once it's reflected, it becomes a negative value. The slope here is represented by Km upon Vmax. Vmax is your maximum velocity of the reaction, right? Now the third factor that we're going to look at is product concentration. We know that the reaction proceeds as substrate acted upon by enzyme to form product. So if your product increases, your reaction is going to proceed in the backward direction, right? Now that means that your the velocity of the reaction is going to decrease. So your enzyme activity decreases. Okay, that is not favorable for the reaction. We move on to the next factor, that is temperature. Let's see how temperature affects enzymes. There is an optimum temperature for every enzyme. We already mentioned that, we established that before, right? So the optimum temperature for enzymes lies between the range of 30 to 40 degrees Celsius, right? This is for most enzymes. Of course, there are certain enzymes like Taq DNA polymerase which has an optimum temperature of 100 degrees Celsius. But maximum of your enzymes fall in the optimum temperature of this range. So your at this optimum temperature, your enzyme activity is maximum. Okay, you increase the temperature, your enzyme activity will decrease. You decrease the temperature, your enzyme activity will decrease again. So on increasing the temperature, we said enzymes are thermolabile, so they degrade, they disintegrate on increasing the temperature. On decreasing the temperature too, the enzyme activity decreases, which is why we say keep food in the fridge, because bacterial enzyme activity decreases and the food remains proper for a longer time, right? We move on to pH. Again, so your temperature basically has a bell-shaped graph, right? We move on to pH. pH, again, there is an optimum range of pH at which the enzyme activity is maximum. What is that range? It is 6 to 8. Alright, here again you have exceptions. For example, pepsin, which has its optimum pH at 1 to 2. Alright, so now your pH again has a bell-shaped graph. On increasing your pH, activity decreases. On decreasing pH, activity decreases. It affects the peptide bonds, right? So there we have a bell-shaped graph. We move on to the next factor, that is activators. Now activators are generally metal cations, but we also have an anion, that is chloride. Okay, chloride is a common anion which acts as an activator for amylase enzyme. Okay, now based on activators, enzymes are categorized into two groups. You have metal activated enzymes and metalloenzymes. In metal activated enzymes, the enzyme is not tightly bound to the apoenzyme. Okay, so it acts as the cofactor. We spoke about that earlier, right? It acts as a cofactor. In metalloenzymes, the metal ion is tightly bound to the apoenzyme and it acts as a prosthetic group. Okay, We move on to the next factor, that is time. When you have optimum temperature, you have optimum pH, you have activators, your enzyme activity increases, it is enhanced, and so the time required to complete the reaction is lesser. That's all that there is to the factor time. Then we have light and radiation. So things like UV rays, gamma rays, other radiations of that sort, they cause enzyme inhibition. Alright, they inhibit your enzyme activity, they decrease your enzyme activity, and that is unfavorable for the reaction. Right? So that covers all the factors that affect your enzyme activity. Alright, so let's start with our last topic for today, that is active site. How does the reaction between substrate and enzyme go about? What I've shown here is an enzyme molecule with an active site. This red thing, the red part that I've shown you here is an active site, right? And along with that, a substrate molecule, right? So now your substrate molecule binds with the enzyme molecule at the active site. It undergoes catalysis and forms the product. That is how your reaction progresses, right? Now this active site has a couple of salient features. Let's look into what those are. The first feature is that your active site is formed due to the tertiary structure of a protein, of your enzyme, right? We already established that the enzyme is a protein. We had seen earlier in our protein video that the tertiary structure formed by the folding and twisting of your secondary structure forms an active site, right? So your three-dimensional tertiary structure is responsible for active site formation. Then we have that your active site is a part of the protein molecule, right? Now protein is formed of amino acids. So your active site is formed by amino acids which in the primary structure, in their linear sequence, are far apart from each other. But due to folding, they come together and they form their active site. Simple? Now these amino acids are called catalytic residue. Okay? The amino acids forming your active site are called catalytic residue. Then we have that this active site is a cleft or crevice or pocket in your enzyme molecule. In the 3D structure, there is a cleft or a pocket formed, and that is called your active site. Alright? Now your fourth feature is that the active site is flexible. It is not rigid, it is flexible so that your substrate molecule can easily bind with the active site and your reaction can proceed. Alright? Then we have the fifth point, that is your active site is formed by two sites. That is your substrate binding site, where the substrate comes and binds with the enzyme molecule, and the catalytic site, where catalysis occurs, where the reaction occurs and substrate converts into product. Right? So these two together form your active site. Our sixth feature is that the coenzymes, we already spoke about coenzymes, right? Those coenzymes which form the enzyme, which are a part of the enzyme, are present at the catalytic site. We already know that coenzymes enhance enzyme activity, right? What is that? Catalytic action. So they are present at the catalytic site. Our seventh point is weak covalent bonds. Now your substrate binds at the active site to the enzyme using weak covalent bonds. Okay. The eighth point is that the active site is very specific. We spoke earlier about how enzyme is highly specific. So that is because of the active site. The active site is specific about the substrate that it binds to because of its shape, because of the amino acids that form the active site. Alright. The last point is very important. Take a close look. So it is about the reaction that occurs at the active site. So what exactly happens? Your substrate comes, binds at the active site with the enzyme. Okay. Together they form an enzyme substrate complex. Okay. They form a complex then at the catalytic site a catalytic reaction occurs and it forms enzyme and product. This product is then released, and this is how at the active site your substrate converts into product, right? So these are the salient features of your active site. So what we've covered here is active site. Now that we've covered these topics, let me quickly summarize it for you. Initially, we started with an introduction about what enzymes are. Then we moved on to nomenclature and classification. We spoke about the 6 classes of enzymes. After that, we moved on to the factors affecting enzymes. The factors affecting it are enzyme concentration, substrate concentration, product concentration, temperature, pH, activators, light and radiation, right, and the time of the reaction, right? And then we spoke about the active site and its salient features. So I hope you liked the video. I hope it makes it simpler for you. Go back to your books and read it now. I'm pretty sure it will become much easier to grasp and retain this knowledge. Alright, see you in the next one.